Hello, welcome back to WIMBA. This is Professor Young, and we are about to begin our journey through the digestive system. As I explained in class, this is truly a journey through a tube, if you would. It is a tube that is connected to the external world, the outside environment, all the way through the entire track as food enters through the mouth. Right on through as it exits the body through the anus. Food is always considered to be on the outside of the human body. One of the things that I do want to just kind of bring to light is, and this is true with anything that we deal with when we talk about anatomy and physiology, and that is how has our understanding of what is happening really evolved over time. And what I mean by that, it comes directly back to this idea of, with the advancement of technology comes an advancement of understanding. And with an advancement of understanding comes an advancement of technology. It's this vicious cycle that keeps propelling the other forward as we go through this. And the digestive system is no different than any other system. As an example, back in the 1800s, back in the early 1800s, there was a gentleman by the name of Alexis St. Martin, who was a trapper, a hunter, and while cleaning his shotgun, shot himself. The rifle, the shotgun misfired. It misfired from a distance of about 3 feet away, and it basically blew a hole into him. And when this hole was blown into him, if you would, it left a permanent hole, a permanent view, if you would, or what we call a fistula, into his abdomen, directly into his stomach. And St. Martin was treated by a army surgeon by the name of William Beaumont, who was first treated by him, and then agreed to actually take care of him. St. Martin didn't have any money. He didn't have the funds. He didn't have the support structure, if you would, at home to go ahead and have anything really be taken care of for him. And so Beaumont agreed to take St. Martin in under the one condition only. And Beaumont would pay for everything for him if St. Martin agreed to this one condition. And the one condition was simply to allow him to do experiments, digestive experiments, within his fistula. You see, Beaumont had this unique ability, this unique opportunity here to pierce into St. Martin's stomach and actually watch the stomach do what it does, which is digest, release mucus, release hydrochloric acid, gastric juices, and watch how all that comes together, that gastric juice comes together to lead to digestion. And so literally, Beaumont would drop pieces of meat, protein, into the stomach and watch as the stomach began to do peristalsis, watched as it would go ahead and release all of these acids and juices into that lumen of the stomach, and watch how it impacted the protein that was put in there. He could go in there with, lack of a better term, a turkey baster, draw out that gastric juice and analyze it for its chemical composition. And so all of this was going on during this time of experimentation on St. Martin as he was recuperating. The hole never closed up. And it was a permanent live, a very rare opportunity for us to be able to really go in and look at what was happening on a digestive level. Today, we have endoscopic techniques. We can drop a camera down into the esophagus, down into the stomach, and actually see how this all pans out. And we'll see a video of that actually in class. So this is an interesting evolution, if you would, of the understanding of the digestive system. As we talked about in class, The gastrointestinal tract, as you should be well aware of, has the task of moving nutrients, moving water, moving electrolytes from the external, from that inner tube known as the GI tract, the gastrointestinal tract, into the body, into the cells, into the tissues, into the cardiovascular system of the body. And the way it does this is through the use of enzymes that are going to break down macromolecules. And really, when we talk about autodigestion, this is what we are referring to. And that is the use of enzymes to break down macromolecules. That's exactly what we're referring to with autodigestion. There is a little bit of a give and take here, because if we release too many enzymes, the enzymes become almost like an autoimmune attack mechanism, in that if the levels of enzymes are too high, it'll actually start to break down the lining of the gastrointestinal tract. There are safety mechanisms that are built in to prevent such an event happening, such as lingual lipase, which is secreted within the mouth from the salivary glands, doesn't do anything in the mouth as far as digestion. It's not until it is activated by the acidity within the stomach that lingual lipase becomes activated. Conversely, if we follow amylase, salivary amylase, which is the enzyme that breaks down carbohydrates within the mouth, salivary amylase becomes deactivated once it hits the acidic environment within the stomach. And so there are mechanisms that are going to control exactly what is happening here. There are about 7 liters of fluid that are produced on a daily basis from the GI tract. This is a combination of water, of mucus, of digestive enzymes, you name it. And about 99% of this needs to be reabsorbed back into the body. Reabsorption of water here is key. We've seen this within the renal system, within the kidneys and the nephron structures. We know that the body is all about that conservation of water. We are a living organism. What that means is, at minimum, about 55% of our body is made up of water, sometimes as high as 75%. The only way that we are able to accomplish that is by a massive effort of being able to reabsorb and conserve that water. And so when we talk about mass balance, that's what we're referring to. High amounts of water going through, high amounts of water being reabsorbed back into the body. The processes that we're dealing with here, of course, we're dealing with digestion, which, as I mentioned in class, is both a chemical as well as a mechanical breakdown of food. And we'll talk about what that mechanical basis is. Absorption, again, going from the external environment into the internal environment. How we move the food down the system, that's all about the motility. And secretion. Secretion is very key here. All right, now, and keep in mind, all of this all goes down to the concept of we have a very unique situation when we talk about the digestive system, because we're talking about taking nutrients from the outside environment, bringing it into the internal through chemical decomposition, if you would, through breaking down complex molecules into simpler molecules. But at the same time that we are doing that, we're also running into the problem of pathogens, bacteria, and viruses that are coming into the lumen of our GI tract at the same time. This is where mucus comes into play. This is where some of the digestive enzymes come into play. This is where the acidic environment within the stomach comes into play. This is where the lymphatic tissue, within the gut is key and critical. If you look at where the heaviest concentration of lymph nodes are within the body, it's right around that abdominal area. And part of that has to do with the constant infiltration of bacteria that we have. Your body has thousands upon thousands of bacteria that are living within it as we speak. They need to be kept in check. They need to be kept in check because if they get out of line, you're going to have a whole host of the fauna, if you would, of your digestive system. Bacteria are supposed to be there. It's okay if it's there, but what's really happening here? And so we're constantly protecting ourselves against that outside world. This here is just a brief overview again of what is really happening here anatomically. We know that we have these salivary glands that are at work. Again, of most importance to us is the parotid. I don't like that color. Is the parotid glands. It's also the sublingual glands and the submandibular glands. Of course, other structures that we are referring to, we're dealing with the esophagus, we're dealing with the liver, the gallbladder, the stomach, the pancreas, the colon, small intestines, colon, the bile duct, cecum, rectum, and of course the anus. All of these structures, and remember, a lot of these structures you are being held responsible for on the cat anatomy. And so you should be aware of that as well and where they are and how they are fitting in to everything else. All right. A couple other things here real quick that I want to mention before I go back to Homer here. Keep in mind that as food travels down this digestive system, as it is traveling down the digestive system, in other words, as it enters in through the mouth and comes down, passes into the esophagus, down into the stomach, and then enters into this convoluted mess that we call the small intestines, into the cecum, into the ascending colon, transverse colon, descending colon, and down into the rectum and out the anus. The food is not always called food. In the mouth, it's called food. As it goes down into the esophagus, it is called bolus. As it enters into the, or as it is in the stomach and is preparing to leave into the small intestines, it is called chyme. And then, of course, by the time it enters into the rectum, it is called feces. And so you have a high turnover rate. It starts out as food, but it doesn't end up as food. And we see that constant changing, if you would, of what is happening there. Also, there's a series of valves that are going to be going on as we go through this, and those valves or sphincters are meant to regulate what is happening. When we talk about these sphincters, we are referring to things such as the esophageal sphincter, which is found as the esophagus is entering into the stomach. We are also referring to the pyloric sphincter, which is going to be at the end of the stomach as it goes into the small intestines. And we are also referring to the, and I can never remember the name of this. There's another sphincter, we'll get to it in a couple minutes, I'm sure. But there's another sphincter that goes from the small intestines into the large intestines. And so all of that is there as well. So kind of keep all that in mind. And, of course, the small intestines are going to be broken down into the duodenum, the jejunum, and the ileum as well. And so we'll go through that a little bit more in detail as we go through this. All right, back to our saliva. I was mentioning a little bit about this earlier. We're going to go into this in a little bit more detail as of right now. Salivary amylase. Right here. This is the enzyme that is going to be released again within saliva, hence the name salivary amylase. And its whole purpose is starch, or, of course, another way of defining starch is simply with the term carbohydrate. And so we have carbohydrate digestion or starch digestion that is happening within the mouth. Carbohydrates are not broken down. They are not digested within the stomach. There's a little bit of carbohydrate digestion that will happen again within the small intestines. And we'll talk again about that a little bit later. But overall, carbohydrate digestion or starch digestion is primarily a activity of the mouth. If you were to take a piece of bread and put it into your mouth and just let it sit on the tongue, eventually, you will get a little bit of a sweet taste that is filtering through there. What that is that starch being broken down into simple sugars. And that's the job of the salivary amylase. Lingual lipase, as I mentioned earlier, is another enzyme that is going to be released from the sublingual salivary gland. And that lingual lipase is there for the purpose of fat digestion. Not in the mouth, though. You're going to have partial fat digestion taking place within the stomach. Lingual lipase is there to do that. It is activated only by the acidity within the stomach. The pH within the stomach is somewhere between a 1.5 and a 3.5. 3.5 is a weak acid by standards of the stomach. 1.5 is a fairly strong acid within the stomach. And so we see that lipase being activated by that low pH level that happens within the stomach here. Mucus, of course, is secreted within the saliva. That mucus is there to lubricate food. Food as it's entering into the esophagus then becomes known as the bolus. Lysozymes are released from the mucus. Again, this is mainly within the parotid salivary gland. But the lysozymes are there to kill bacteria. All right. The lysozymes are there to kill bacteria, and so they kind of secrete, if you would, or coat the overall mouth. Immunoglobulin A, again, this is going to be one of our types of antibodies within the body, is there to inhibit bacterial growth. And we also have some electrolytes that are found within saliva as well. Sodium, potassium, chlorine, phosphates, and bicarbonate, HCO3-. That's your bicarbonate there. And so all of these things are found within saliva. Saliva, in particular salivary amylase, is very happy within the mouth. It functions on a pH of between 6.8 and 7. And so it's very happy. It's very comfortable in that environment. Again, lingual lipase, not so happy, not going to activate. Once it gets down into the stomach, salivary amylase is deactivated. Lingual lipase kicks into high gear. We've talked about this a little bit. I'm not going to spend too much time on this. But we know that the saliva is produced within three main glands: the parotid gland, the submandibular gland, and the sublingual gland. They are, of course, highlighted right here on this little diagram. There is the parotid. Here is the submandibular, right where you would expect it. And then underneath of the tongue is the sublingual gland. And so all three of them guys are sitting right there. The stomach, as we move down, and then I know I kind of skipped over the esophagus here. I kind of did that on purpose only because there's not much happening within the esophagus. What I will say is this. We have muscular control over the esophagus for about the first inch or two. In other words, we have enough control over the esophagus to get the food down into the esophagus. Once that bolus is 2 to 3 inches down into that esophagus, smooth muscular contraction is taking place. Smooth muscular contraction is taking place. And that smooth muscle contraction that we are referring to, that takes place within the esophagus, that takes place within the stomach, takes place within the small intestines, takes place within the large intestines, is what we call peristalsis. So peristalsis is a wave of muscular contraction within smooth muscle of the GI tract, of the gastrointestinal tract. And of course, you can't see that at the very bottom, but it's there. So it's a wave of muscular contraction within smooth muscle of the gastrointestinal tract. And again, what happens here is the esophagus, which is coming down right into this area, by means of the esophageal sphincter. All right, so the esophageal sphincter is what is going to connect the esophagus into the stomach. The stomach is made up of 4 main regions. Those 4 main regions are the cardiac region, which is going to be right here. So aptly named because it is located pretty much in front of the heart, or at least where that esophagus comes in is also related within the heart. We have the fundus. Fundus is basically extra storage area within the stomach, and so once you have stuffed yourself with that big old Thanksgiving dinner, and you think, I have just a little bit of room to go back and shove in some more food, it's your fundus that's going to be overflowing. That's where all the extra food goes. You have the body of the stomach. And the body of the stomach is going to be right here in this region. And then you have your pyloric region. And I know this is going to overlap a little bit. Don't yell at me. Don't scream at me. Don't ask questions. But you have your pyloric region that is kind of right there. And it overlaps a little bit with the body. The body's actually up a little bit more. But throughout all of this, we have the gastric rugae. And the gastric rugae is a series of muscular ridges. They are more prominent when the stomach is empty. They are less prominent when the stomach is full. And the reason for that is because your stomach has expanded. And these ridges of smooth muscle also expand out to allow the stomach to accommodate that third helping of turkey, the second helping of stuffing, your fourth piece of cranberry sauce, and your sixth big old scoop of mashed potatoes along with your fourth biscuit. That stomach is going to expand. And so the smooth lining or the rugae, that muscular ridges that are normally there, expand out, and they are less visible when the stomach is full. More visible again when the stomach is empty. And so that's pretty much your overall structure. What I will also point out is you have, and this is kind of obvious here, it's not really though, but the outer edge, if you would, of the stomach is what we refer to as the greater curvature. It's called the greater curvature. And then your inner, right there, is what we call the, you've probably guessed it hopefully by now, the lesser curvature. All right, and so that's kind of the overall anatomy. In lecture, you have seen or you will see peristalsis at its best. Peristalsis, again, is that wave of muscular contraction that is going to happen through here. And as this begins or as this happens, that food is tossed around and it's mixed with all the gastric juice. We haven't talked about that gastric juice yet. We will here in a couple minutes. But that gastric juice swirls around and mixes around within that bolus that is in there. And that helps to make sure that we coat everything in there to aid in digestion. The other thing it does is it bounces things against the wall and against each other to help to break it up. That's the mechanical aspect of that. We'll see that in the video. It's a very good view of what exactly is happening there. Cells of the stomach. We have 4 main cell types that are present within the stomach. These 4 main cell types have the distinction of secreting various types of substances, from mucus to hydrochloric acid to a variety of enzymes. And together, these 4 cell types produce, and this is literally the term, it's not because I can't think of another term to call it, this is what we call this, that mixture of everything that's released by these 4 cell types is referred to as gastric juice. And we produce about 2 to 3 liters of this gastric juice on a daily basis. Some of this is going to be fairly obvious to you. Some of this, you're going to sit there and say, well, geez, that makes sense. Others of these you're going to want to make sure you pay attention to because they're not as obvious. One of the duh moments are mucus cells. Mucus cells line the stomach, and their purpose in life is to secrete mucus. It's what they do. Why do we need mucus? Well, the mucus is there to help to protect the lining of the stomach, the smooth muscle lining of the stomach, from the intensity of the acid. And so it helps to neutralize the acid right along the edge. Otherwise, that acid would begin to eat through the lining of the wall of the stomach, and you would have yourself a ulcer, which is not good. Keep in mind that the majority of stomach ulcers are actually not caused by an overproduction of hydrochloric acid. It's not caused from a lack of mucus, although that can be contributing factors. There's a type of bacteria called Helicobacter. Helicobacter pylori. And it's also what we call, it's abbreviated as H. pylori. It's that bacterium that is actually the cause of the majority of the stomach ulcers that we see within the United States, and that actually affects the mucus of the stomach, and then allows that hydrochloric acid to get in there and really begin to eat away. So if you get rid of your Helicobacter, you solve your stomach ulcer problems. Parietal cells. Your parietal cells are there to secrete hydrochloric acid. They're also secreting intrinsic factor and ghrelin. Now, pay attention to this. All right, pay attention to this. Ghrelin is actually one of these guys that is involved in hunger reflexes. Ghrelin induces or promotes hunger. Ghrelin is going to induce or promote hunger. And we see it released from within the fundus, the extra storage area, if you would, oddly enough, of the stomach. All right, and so that's what we see going on there. Intrinsic factor, we're going to see involved a little bit more later on. It's released within the stomach, but it actually has a lot of its impact within the small intestines. Intrinsic factor is going to bind to vitamin B12. And as it binds to vitamin B12, it allows it to be absorbed easier within the small intestines once it gets into there. Vitamin B12 is a key vitamin involved in the synthesis or the production of hemoglobin. And so you want your vitamin B12 levels to be normal or at normal or above because of that hemoglobin factor. So we've got hydrochloric acid. Hydrochloric acid is important because it's going to activate lingual lipase. It's also going to help with the prevention or the blocking, if you would, or inactivation of salivary amylase. Hydrochloric acid is going to help to also break down the protein structure in general. Parietal cells are going to release that intrinsic factor, bind to vitamin B12, be able to carry on with hemoglobin production, and ghrelin, that hormone that's going to promote hunger within us. For a lot of people, if you overproduce the ghrelin, you have a hard time of controlling that appetite. They always say you need to eat for at least 20 minutes. And if you're one of these people that sit down at a meal and you eat it in 5 minutes and you're still hungry, what's happening is ghrelin is still being released. Now, you got to remember, your stomach, your digestive tract, has its own nervous system, the enteric system. You probably remember that a little bit from, hopefully, A&P1. The enteric system is a nervous system associated with the stomach. Ghrelin is going to be released, which is going to activate or act upon the enteric system, sending a message to the hypothalamus, which is going to say, hey, anyone home? I'm hungry. It takes a while for the enteric system to realize, wait a minute, there's no more ghrelin being released. Slow reacting. It's slow acting here. And so that's why they tell you need to eat for about 20 minutes because that's when the effects of ghrelin wear off on the enteric system. Kind of keep that in mind. If you can control the release of ghrelin and if you can slow down your eating habits and take that one plate of food and eat over a longer period of time, you're good to go. Sheep cells secrete enzyme pepsinogen and gastric lipase. What does all of that mean? Enzyme pepsinogen, I want to see something here real quick. Okay. We're going to talk about a little bit on the next slide here underneath the digestive enzymes. We'll talk about pepsinogen. Pepsinogen actually is cleaved, if you would, from hydrochloric acid. And so sheep cells are going to release the pepsinogen, the enzyme. It's going to be cleaved by hydrochloric acid. It's going to produce pepsin, and pepsin is basically helps to digest the proteins that are found within the food that you are eating. Here's the other thing, though. Pepsin also helps to cleave more pepsinogen. And so as pepsin is released, the body releases more pepsinogen. Pepsin is going to cleave pepsinogen. Now, the reason for that is to actually reduce the overall levels of hydrochloric acid. Hydrochloric acid starts the process of cleaving or shortening or activating pepsinogen. Pepsin then has that auto cleaving ability within it. And gastric lipase. is also released here. Gastric lipase, again, is going to be an enzyme that is going to help to break down or catabolize, if you would, fat within the stomach. Again, the majority of your fat absorption and digestion is going to really take place within the small intestines. And then your enteroendocrine cells, they're going to help to regulate your overall digestion process. And so you need to know these guys all. Specifically, make sure you pay attention to the mucus cells. Definitely pay attention to parietal cells and also your chief cells. I want to, I'm not going to do that right now. All right, let's go on to digestive enzymes. Enzymes are typically released as inactive forms of the enzyme. Those inactive forms are what we call zymogens. The reason for them being released as an inactive form is that they're only going to be activated when they are needed. Otherwise, they're going to cause damage to the tissues. If you have all of these enzymes that are all activated all at the same time, all the time, they're going to start eating away and again, have that autoimmune impact or effect, if you would, on the lining of your GI tract. You don't want that. You don't want that. You want to make sure that whatever is in that GI tract is self-contained until it is ready to be absorbed. To help that, we release these enzymes in an inactive form, or we have the ability to deactivate them at a later time. Again, prime example, lingual lipase, absorption of fat. Not until we're ready for it, though, because otherwise it's going to start to break down the fat that you have naturally within your tissues. You don't want that. We need a certain percentage of fat. And so that's why it's released within the mouth, but it's not activated until the stomach. And as soon as it gets into the small intestines, it's deactivated. And so it's self-contained in where it can go ahead and work. What does that? In this case, it's pH. All right. Now, pepsinogen is no different. And I was explaining this on the slide before. Pepsinogen is no different here. Pepsinogen is released within the stomach. Again, it's going to be released by the chief cells. And hydrochloric acid activates pepsinogen. It cleaves the amino acids. It shortens the chain of that enzyme and creates pepsin. Pepsin is the enzyme that is responsible for the breakdown and the digestion of protein. It's going to break it down into smaller pieces. It's not complete digestion. Not until we get to the small intestines. And again, it's what we call autocatalytic. It has that itself ability to break its own self down. And so pepsin will go ahead and actually break down and cleave pepsinogen into pepsin. Why? Again, it goes back to restricting the amount of hydrochloric acid that we have released at any one point in given time. Keep that in mind. Gastric lipase, again, it's going to work with lingual lipase to break down fat. It's going to surround the fat. It's going to surround the fat, and it's going to help to emulsify it, to help to break it down into tinier fat goblets. So that way it's an easier time of reabsorption within the intestinal tract. Remember, the lining of almost everything here of your cell membranes is that phospholipid bilayer. It is hydrophilic on the outside. Fats are hydrophobic, and so it needs a way to get in there. That's what's going to happen here. And so gastric lipase is going to work with lingual lipase to do just that. Surrounds those fat, breaks it up into more fat globules for easier time of reabsorption within the small intestines. And then we have intrinsic factor. Again, that's going to be helping to bind to your vitamin B12 to get that into the intestinal tract a little bit easier or absorbed. Peristalsis. We're going to see this regulated within the greater curvature. As I've mentioned already, it is a wave of contraction. It is a wave of contraction. Within the stomach, it's going to start at the fundus, and it's going to work its way posteriorly down the stomach. And as I've already mentioned, the function of this is to swish up the food, if you would. It mixes the food or the bolus in with the gastric juice to help to break it down, and it's going to also aid in physical or mechanical digestion, the physical act of breaking everything down. In class, we looked at the video, or we will look at video of the peristaltic wave of muscle contraction as it goes down into the stomach. And so we'll look at that at a little bit later of a time. I can't do that through WIMBA. Typically, during the height of digestion, we have one contraction every 20 seconds. I think now is a good time. I'm going to stop this session of the WIMBA, and I'll be back with you here in a few minutes, and we'll go through and split this up into 2 WIMBA sessions. And so we'll go through the rest of the digestive system here in a couple minutes. Again, review this. If you have any questions, always feel free to shoot me an email or ask me in class, and we can make sure that everyone is good with this. My ultimate concern is that you guys learn this and you know it, you live it and you breathe it, and you actually gain an understanding. You're not just memorizing facts. And so good luck with that. I'm here if you need any questions.